So hopefully you all recognize these structures: nuclear membrane, mitochondria, Golgi apparatus, and you notice that all 6 of them are listed and all 6 are described. And you still have your cell projects with you, so these should really be not a lot of thinking. There's two of these are an issue, and the 2 that people got confused, I think, are A and D, and we'll look at those a little closer than the rest of them. And I promised you after the last cell test that you would come back and review it, and that's what we did, right? That's what you were supposed to do yesterday in class. The first one says active in synthesis and sorting, secretion. So what do you think? Active in synthesis, so making something, sorting, and secretion of cell products. Golgi apparatus would be my first guess too, because of this word secretion, right? We're thinking, man, the Golgi apparatus is involved in secretion, right? So that's kind of like, that's a big clue. Yeah. Yeah, could you turn the light on? So anytime we're talking about sorting, so the Golgi apparatus is the post office of the cell, right? So anytime we're talking about secretion and packaging and sorting, we're thinking Golgi, all right? So digestive organelle, I hope that's easy for everybody. What is it? Lysosomes. Really easy. Organelle, where most cellular respiration occurs, you make it most of the ATP or all the ATP in the cell. Where is it? Most of the ATP. Where is it? Mitochondria. Notice I said most, and we'll go into that when we do cellular respiration next week, photosynthesis the next. Active in membrane synthesis and synthesis of secretory proteins. Proteins that are going to be secreted. Rough and smooth ER, that's right. So remember that in the smooth ER we make membranes, we make lipids. And what are membranes? Lipids, right? So it's a smooth and rough ER. We could just put endoplasmic reticulum ER, right? That includes the rough, where we make what do we make in the rough? Does anybody remember what we make in the rough ER? Ribosomes are on the rough ER. That's why they're rough. You're right. What do ribosomes make? No? What do ribosomes make? At least you're thinking biomolecules, so that's good. But proteins. So ribosomes make proteins. You see, that's the key, right? Secretory what? Proteins. So the rough ER is making the proteins, where the smooth ER is making the what? The membranes or lipids. That includes cholesterol and other lipids. Active in lipid and hormone synthesis and secretion, in a sense. Lipid and hormone synthesis, smooth ER. Photosynthetic organelle that converts light into sugar. Chloroplast, that's an easy one. Explain how the membrane surface area is increased within cells or within cells organelle. So you have this tiny little organelle that's inside this tiny cell. How do we increase the surface area without making a mess? Having to do what now? More materials go into the cell. So do you understand what surface area is, I guess is the question. Is that, I don't know, or what is that? Is that an answer from anybody? We don't know what surface area is? Okay. Let me pause this again. So we don't understand what surface area is. That's my understanding. So that's what I'm hearing, right? Is that right? Okay. So when we're looking at a cube, let's look at a cube, because that's pretty much something everybody can imagine. You can imagine a dice, is that right? I could draw a basketball too, right? But the problem with trying to draw something round is, for me anyways, it's hard to kind of imagine that there's 3 dimensions there, right? Because all you see is the circle. So with a cube, it's easier to draw. It's not quite a cube because I messed up, but let's go ahead and think of that as a cube. So if we're looking at area, who knows what area is? The inside? Did somebody say inside? What is area? From math, length times width, right? So length and, pick one. So let's pick one side. I'm going to number these. 1, 2, 3. There's a 4, right? And a 5, right? And then there's a what? A 6, right? So there's length. Each side has a length and a width. If I multiply those two, length times width, that equals area. Do you understand that? Yeah, that makes sense, right? So what do you think surface area is? What? All the areas where? On the outside. Very good. So 1, 2, 3, 4, 5, and 6. So all of those put together are the surface area of the cube. Does that make sense to you? I see 3 people nodding yes. But what I would love to hear is these people participating and saying, yes, I understand. Or no, I don't. Please answer the question. Answer my question. Are we good? No. Okay, I see some no's, and that's okay. You don't have to say who you are. So, can anyone, because it looks like some people are shy, can someone tell me what somebody might be confused about that? What's confusing about that? What is confusing to you about what you're seeing in front of you? Yeah, go ahead. That's a good question. What would you do to find the surface area is what you're asking me, right? I'm not asking you that, right? But the answer is easy. I'll tell you in just a second. But I'm not asking you to tell me how to calculate it, right? That's something that I would do if this was a more advanced class, really. It's not that advanced, but I'm not going to ask you to do that. In fact, but I do ask you to do that on one of the activities, don't I? Isn't that one of the activities to do that you were supposed to do? Or are you getting that tonight in the packet that I'm about to hand out? I think that's tonight's. Yeah, you're going to have to calculate surface area. You'll learn more about that today. But, so you found the surface area of one side. It's a cube. Let's pretend it's a cube. It's not really a cube because you can see it's not really square. It's more of a rectangle. But let's assume that this is a cube, right? And so all sides are going to be the same. Do you agree? So I find the area of one side, then what am I going to do? I'm going to add what? This area, and then this area, and then this area over here, and then that area over there, and then an area underneath, right? I could, because they're all the same, or I could add each of the areas together. Either way gets me the right answer. Do we all agree on that? So the surface area is what someone said already, that it's all the area on the outside of the cube. Right? I mean, I think that's pretty clear. Do we understand it that far? Okay. So now let's put that away. And let's ask, if we then have this organelle, and I want to increase this organelle's surface area. And what do I mean by that? Why do I want to increase surface area? Let me go back for a second. Why is surface area important for a cell? Let's assume this is a cell, it's a cube cell. What's important about surface area? Could you turn that fan off? It's right in front. Turn that down. It's in the middle. Thank you. Say that again.
The cell's surface area can change if it's shrinking or...
Well, that's true. If it shrinks and expands, it could change the surface area. But there's a more important question. What is the surface area helping with? What's it going to do? What happens at the surface of a cell or an organelle? Well, let me redraw it then. What's happening at the surface of this? So there's a cell. What do we call it? Oh, it's doing it again. What do we call, oh, it's the pencil. What do we call this? This part. So, right, so what is it? So this is the membrane. So what's happening at the membrane? What are the functions of the membrane? That's like the whole point of this activity, right? What is it? Letting things in. So there's a plus, letting things in. And what? And letting things out. So things going in and out. That's the purpose of the membrane. Can you imagine for just a second that the more surface area you have, the more stuff can get in and out? Does that make sense? Now we're talking about things going in and out by something called diffusion, right? All right. So diffusion goes from high to low. Can you understand that if I have a cell that's I'm gonna have to, my pencil's dying, so I think that's the problem. I thought it was the iPad, but it's the pencil. So let's take a look at a cell that's this small and a cell that's really humongous. All right. If things are diffusing, which is random movement, right? It's not, so it's going to take time. And there's some part of the cell over here in the center that needs oxygen. So oxygen's out here, O2, and it needs to get to that part of the cell. Are we good with that? All right. I see people doing their hair. I'm sure it's important, but I really would rather you focus on what I'm saying here. So this oxygen needs to go from here into this middle part of the cell. If it's which one, and let's say the same cell, there's another cell over here, it's much, much bigger, and there it is, the same part of that cell. That another oxygen molecule has to get to that center. If it's going to diffuse, how long do you think it's going to take for something to diffuse from this part to this part? Not very long, because it's a very small distance from the membrane of the cell to where it has to go. The bigger the cell, the longer it takes to get there. Do you see that? Okay. So there's going to be, so what we call this is the surface area, SA. Can I just use SA? Surface area to volume ratio. There's a surface area to volume. So there's a sweet spot where you get the maximum amount of speed and still the cell can hold the most amount. But if you go beyond that, if that number gets too big, if the area too small, because the volume is really big but the surface area is small relative to that, if the cell gets too big, then the cell can't function. That's why there's a size limit on the cells. Does that make sense? All right. So there's a surface, just because it takes time for things to diffuse. If it takes too long for something to go from this end of this edge of the membrane all the way to the center, then that cell can't do its job. Huh? I can't hear you. Something's going on outside with that. Good question. It all depends on the reaction, right? So we're not really talking about hours, we're talking about seconds. Things happen very, very quickly in living systems, don't they? If you have a thought in your brain, how long before if you say kick the ball, that's the thought that you have in your brain. How long does it take you to kick that ball? A split second, right? If you're a good athlete, it takes you a split second. You have a thought about kicking the ball, you kick it, right? Right? Does it take you an hour to figure that out? There's a lot of signals that went on there. You involved 5, 6, maybe 100 cells. and they all sent signals, and you sent a message all the way from your brain, all the way down to your leg, just like that. So imagine that. You go to touch a hot iron, and it's hot, you burn yourself. How long before you pull your hand back? Like less than a split second, right? Even though it doesn't go all the way to your brain, so you move before your brain figures it out, because your brain gets a signal after you're done. But that movement was from the surface of your skin to the nerve, then to the second nerve, to your spinal cord, then back to the nerve, to the muscle, and then the muscle contracts. All that happened in that little split second. So these reactions have to be very fast, right? That's all osmosis and diffusion and some cell signaling, but it's all very quickly done. So these concentrations and these speeds are amazingly quick. But that's only because your cells are in this sweet spot. If they were too big, then they wouldn't work. Question.
Okay, so if something happens and then like your brain don't get alerted right away, is that why it hurt after the fact? After it happens?
Yeah, to some extent, there's all kinds of reasons why that happens. See, what happens is that what we just talked about was a reflex. And so when you have a reflex, your brain's not involved in it. So, you'll sit on the doctor's table and they'll take a little rubber hammer and they'll hit your knee and your leg moves and you didn't even move it. You didn't think about moving it. It just moved. That's called a reflex, all right? Somebody tickles, touches the bottom of your foot. Before you know it, you don't know this, but before you know it, your toes curl up. Then you start feeling the tickling, right? So you feel there's a delay. Your brain finds out after you moved. That's called a reflex arc. But still, it involves many cells and very many different signaling mechanisms. It still involves osmosis, diffusion, facilitated diffusion, and all that. Okay? So we're good with that? That's one of the membrane's functions, is cell signaling. That's one of the questions on the project that you had yesterday. Okay. So that's surface area to volume ratio. So now my question is, and the question in the package is, here is this organelle, and it wants to increase the surface area so it can move more things in and out. What is it going to do?
Create more of a membrane?
Right, but it only has so much space. You have the inside of the cell, right? So here's this cell has a nucleus, right? Has a nucleolus, right? Everybody agree? Has a Golgi body. Endoplasm is particularly rough, a reticulum rough and smooth. Has a Golgi body, kind of something like this, right? And has lysosomes and mitochondria and whatever, right? Do you agree something looks like that? And we know this is the plasma membrane here, and that's the nuclear membrane here, and it's badly drawn, but we got the idea, right? So now the question is, how do you get something like the endoplasmic reticulum? It has to do a lot of stuff, it needs a lot of surface area because it's making a lot of proteins, it's sending them, it's making lipids, it has to get rid of them. It has a lot of stuff, needs a lot of surface area, but only has this much space. It can't take any more space up because the nucleus is there, over here some mitochondria. So it doesn't have a tremendous amount of space, it's very jam-packed in there. How do you increase surface area without increasing volume? Remember surface area volume? How do we increase surface area but not increase volume? In other words, we keep the amount of space the same, but we increase the amount of surface area available. Well, we could get rid of stuff that would increase the amount of space, but how do we do it without increasing the amount of space the organelle is taking up? So take a look at a sheet of paper. Take out a sheet of paper and look at it. That's it. This paper has a surface area, doesn't it? Yeah. Okay. How can I either decrease the volume or increase the amount of surface area without increasing the volume? I fold it. That's right. We can fold it. So if we have this thing, let's say it's this long. This is how much space we have. And we want to have more surface area. Well, what can we do? We could just do this. Same space, look at it. It's the same space, isn't it? But now we have what? A lot more surface area. Well, before we just had this much, now we have this. That's a lot, isn't it? It's a lot more. Every fold gives it a lot more. What's an example of that? Well, if I told you to walk a mile, if we had a line that was a mile long, and it was a straight line, so it's a straight line from here to here, 1 mile long, where somebody else, I told them to go this path, which path would you want to go? This path from A to B, or would you rather walk this path from A, and I'll just hurry up and end up at B? Which would you rather take? The straight. Why? It's shorter. So what are you doing if you're increasing the distance from A to B by going up and down, folding it, in other words? What are we doing? We're increasing what? Surface area, because we increased one of the dimensions. So if this is equal to 1 mile, but now this is equal to 1, 2, 3, 4, 5, 6, 7, now it's 7 miles, even though you're still going from A to B. So in other words, your volume, how much space you're taking up is the same, but we've increased the surface area in the same amount of space. Is that clear? So that's what we mean by folding. That's why folding is the answer. That's why your brain surface area is folded. And now I'm going to talk about another organelle. And this is going to take up pretty much the rest of today. So one of the things that we do, and you've all of you have seen zombie movies, right? Or monster movies where somebody rips open somebody's gut and out come their intestines, right? There's all this disgusting intestines just flowing out, or blah, 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 right? This is disgusting. But what happens is, so that tells you something about your intestines. What are your intestines? They're folded, that's right. Why do we fold our intestines? There's more space without making you a giant, right? Because you'd have to have a giant belly to have the same amount of space. If it was not folded, you need to be about a couple 100 feet long, right? That wouldn't work for anybody. So why do we need that much space? So let's talk about that. That leads us to a real big problem that I noticed in first block. I've said it many times, but I don't think people understand it. So we're going to go ahead and talk about it. So here's this thing called, the stomach. It's going to look something like this. It's not well drawn. Okay, so let's just can we not pick on me today? So it's not well drawn. And so this area of the stomach has a volume, and it keeps going, and then it turns like this, right? That's your intestine. It's much better packed. You're packing it much better, okay, in a living system. But let's go ahead and draw the tubes. And what's at the other end here of the intestine? Guys, what's at the other end? One side of the intestine is called what? Really? One side's called the mouth. You know this. Everybody knows this. This is the mouth. This everybody should know. This is the mouth. And what's on this end? It's the anus. It's not Uranus, it's anus. Well, it is your anus, but it's not Uranus. Uranus is a planet. That's why we pronounce it Uranus. There's a colon and small intestine. Those of you that went to the anatomy camp saw that. There's a colon, small intestine, then there's a colon. And so what's the point of me drawing this? A lot of people in this room still believe that when you eat something, when food goes into your mouth, right, down your throat, called esophagus, and into your stomach, that's inside your body. Just close the door, please. You think that's inside your body. It's not inside your body. It's still outside. I've said this many times, haven't I?
Yeah, that we're a tube inside a tube.
You're a tube within a tube, yeah, that's right. I've said that many times, you're a tube within a tube. So people don't understand it, so I want to spend some time trying to make sure that you have a good chance at understanding this, that this blue stuff is still outside your body, even though it's in your stomach. It's inside your stomach, but it's still outside your body. So what's inside your body? This part is inside your body. This part right here. So this is where your blood, your heart, your muscles, your connective tissue, that's all this green stuff. Do you see that? This green stuff is inside your body. This is where your blood vessels are, your arteries and your veins. But inside this tube that you call, that we all call intestines, right? It's a tube. And then, of course, that's on both sides, right? So there's a tube. That means that inside, and this is a cross section, so that means we've cut this person open, right? So we have this tube. And on both sides of this tube is this stuff that we call skin, right? Do you agree? And on both sides is we have this thing, this stuff that's called skin. On this side, that creates the tube on the inside. Now you may not remember this, but I'm sure you remember me saying to you that if we could, we cannot do this, but let's think of something, let's say we could. If we could swallow a piece of floss, right? I said, somebody remembers me saying that. Swallow some floss, have it tied out, and you would have it come out the other end, and we could floss ourselves, right? It would hurt very much. You can't do it for various reasons, but if we could. What's on the other side? This is skin. So this is the outside of your skin. This is your lips and your hand and so this is your lips and your hand and your outside of your throat, right? It's all out here. And your stomach and your legs, right? And your butt is all out here in this part. And so this is your head, right, out here. Now I'm not drawing I'm obviously not anatomically correct, right? So this is your butt, and this is your butt cheeks, and so I'm not drawing it anatomically correct, but that's not really the point, right? It doesn't look like a human. It's not supposed to look like a human. What am I trying to make a point of? That you are a tube within a tube. We are very much like worms. And if you ever looked at a worm has a hole, right? And then there's another. And then if we cut the head off the worm and the tail off the worm, we'd see something that looks like this, right? And it looks something like this. Do you agree? And then it has an inside. This is inside the worm right here. So when the worm eats dirt, the dirt goes where? In the hole. But then the stuff that it wants, the nutrients that are the organic material, the minerals that are in the earth that the worm wants, the worm has to do what with it? Has to bring it into the green spot, the green area. Do you see that? So you see why we call it a tube within a tube? There's a tube, this is a tube, and inside that tube is what? Another tube. And you're the same way. You have a mouth that goes throughout your body, that's one tube, and it's surrounded by another tube. That's that green stuff. That green stuff is your eyeballs and your brain and your heart and your muscle, all this stuff that's in your blood, all this stuff that's actually in your body. So when you take in food through your mouth, it's not really in your body. It's in that inner tube, that tube that's inside of you. And it's continuous. Do you see it being continuous? Now, why don't you just fall in half, right? Somebody might say, well, why does this half fall this way and this half fall this way? Because it goes all the way around. We're just cutting it, right? So if we took you and cut you in half, this is what we would see. We'd see a tube inside a tube. Do you see what I'm saying? Okay, so when you swallow stuff, now think about it. This is what makes everything make sense. How is it that you can have you have bacteria in your gut? You know that, right? We've talked about it. We have some bacteria. Let's make them red. They're just in your gut. They help you very much, right? They help a lot. Without them, we couldn't digest some of the things we digest. In fact, they explain farts. Without them, we wouldn't have farts either, but we couldn't digest half the food we digest. There's more of them than there are us. There's more bacterial cells. They're smaller, but there's more of them than there are our cells. inside our gut, inside the tube, which is outside, right? And sometimes, yeah. And outside you have, on your skin, you have what? What do you have on the outside on your skin? I hope everybody understands you have on the outside of your skin. Bacteria. You have bacteria, you have fungus. You know, you've heard of yeast infections. Those are fungal infections. Yeah, there's a whole little life, there's a whole ecosystem on the surface of your skin. And they're everywhere. They're in your stomach, they're in your throat, they're in your mouth, right? So those bacteria are all outside your cell. Even though they're inside your mouth, the stuff that's inside your mouth is not inside you, right? This stuff is not in your mouth. It's not in your body. It's inside your mouth, which is where? It's continuous. See, look, you can put your finger right in there. Can you put your finger into your heart? No, because your heart's inside your body. But you can put your finger in your mouth. Why? Because it's still outside. So here is all these bacteria. They're all over the place. They're fine outside. They're not a problem outside. It's when they get where? Inside. If they get to this green part, you got problems. You got what's called septicemia. Septicemia. Write that down. Because some people, God forbid, but some of you know might actually, I'm going to tell you right now, I'm going to put all this on the test. I'm guaranteeing there'll be at least 2 questions on the test. And you're going to have to describe what's inside, what's outside, how does stuff get in. I'm letting you know right now.
How do you spell it?
Septicemia. Anybody know how to spell it? Septicemia. Septicemia. I can't, it's hard to write with my finger, I apologize. It's messy. Hopefully the pencils be charged in a second. So septicemia, and again it's Septicemia, is when blood gets infected with bacteria. When these bacteria that are fine outside on the skin, and they're fine on the outside here in your intestines and your stomach, everything's fine. But when those bacteria get into the green part, they go crazy. You are really good food. You're excellent food. You're full of a lot of minerals and vitamins and fats and proteins and sugars. You're ready. You're everything a bacteria dreams about. All the food that they need for very, very many generations. So the only reason you're not being eaten right now by them is because you have something called skin. And skin protects you from those bacteria. Very tightly organized cells that act as a barrier against them. Those membranes protect you from these other more destructive organisms. Do we agree? Does that make sense? Does anybody have a question or problem? Yeah.
So what about when you get like cuts and stuff?
Well, what happens when you get a cut? What are you worried about if you get a cut?
Infection.
An infection. So what's an infection? The bacteria coming in there and attacking you. That's why you're worried about it. In the old days, that's what killed most people. Most people died before they were 30 or something like that. Why did they die before they were around 30? Because of the infections. In fact, one of the biggest sources of infections was here in the mouth. Toothaches were one of the biggest sources of people dying. Doctors were, the first doctors in the Old West, one of the big things that they were dentists. One of the most important things people went to go see. Oral hygiene was incredibly, it still is, and people don't really recognize it. And some people think that heart attacks, one of the sources for heart attacks, is bad oral hygiene. Because you get cavities, and what happens is bacteria get in that green part, and they get lodged in your heart, and they cause all kinds of damage. So
when you get a cavity, it's like another opening.
a cavity is created by bacteria. They are, the acid from the bacteria are eating away your enamel. That's right. That's why it's bad to feed, because what is sugar? Food. For who? For you, but also for who? Bacteria. And they love it. The more food they have, the more of them there are. Huh? What'd you say? Yes, I know. Yeah.
So, as a So your digestive tract, the bacteria is stealing nutrients from the food, and you're also taking nutrients from it?
That's an excellent question. It's what we have with bacteria is a symbiotic relationship. Could somebody write that? Could you all write it down? I know there's some conversations going on. Let's call it a symbiotic relationship. And it's something that we like. It's a type of symbiotic, just means close, really close relationship. Symbiotic. Do you all know how to spell that? Let's hope my pencil's charged already. Symbiotic. Symbiotic. That means symbiotic means close relationship. I don't think you can get much closer than being inside the tube that, in your stomach or in your intestines. They grow up with us, these bacteria.
Isn't a symbiotic relationship when both benefit from it?
A lot of people think that, yeah, it's a confusing point, but no, the answer is no. A lot of people have this misconception that symbiotic means you're both benefiting from it. That's not true. Symbiotic does not mean you both benefit from it. Symbiotic means you're just a close relationship. I wonder why that happened. That's so weird. Now, what is a relationship where both hurt? Well, what are some of the concepts? What are some of the symbiotic relationships? You have parasitic. Parasitic,
One feeds off of the other.
where one feeds off of the other. Do you all agree on that?
Yeah.
So parasitic. Let me spell that correctly, that would probably be helpful. That's where one feeds off the other. So one negative, the other what? Positive. One positive. What else happens? What other kinds of symbiotic relationships might you have?
I can't remember the name of it, but both of them are just sort of there.
They're both sort of there. Why don't we give that as a homework assignment so we can move on? You guys come up with the other types of symbiotic relationships. And let me tell you this, that in this case with bacteria, both of them, both you, this is you, right? The green part is you, so is the black. The black and green part's you. You, and this includes all animals, right? Because all animals have this symbiotic relationship. And the bacteria benefit. Both benefit. They're both, this is a plus for the bacteria and it's a plus for you. Without those bacteria, you couldn't survive. You could not digest most of the things you digest. Digestion means breaking it down. So you couldn't, we need these bacteria to help us break down. One of the first things babies start doing is they start putting things in their mouths. What they're doing is they're starting to collect what? Bacteria. Because when they're born, they have no bacteria. That's why their poop is so slimy and ill-formed, right? And they just don't have the ability to digest a lot of stuff. That's why moms, one of the first things that you ate when you came out of your mom was lactose. And that's pretty much the only thing you eat. It's mother's milk, right? That mother's milk has everything you need to grow initially in the first few months. And the reason you need that digested for you and produced is because you have no bacteria to help you digest all that. Yeah, question?
So if the bacteria gets into the green part, would it become, instead of positive, negative, because it's hurting you?
Right, it no longer is a symbiotic, it now becomes a predator-prey, or it becomes an opportunistic infection. So that relationship is changing, right? But as long as everybody's staying on their side of the membrane, right? Then how do the bacteria benefit? They get food brought to them. You serve your bacteria every day, right? You go and you work hard, you go get a sandwich, you put it in your mouth, you chew it up for them. You chew it up for them, you throw it in your stomach, you put acid on there for them, and then you go ahead and you digest, you break it down, and they help you break it down. They eat a lot. But a lot of it they don't eat. What happens to that stuff that they don't eat? What's the whole point here?
Release it from your body.
Well, the stuff that you don't eat and they don't eat, where does it come? Where does it leave? It leaves, yeah, your back door comes out here, right? Right? But the stuff you are able to digest, what do you have to do? There's one other thing you have to do after you broke it down. What do you got to do? Get it to you. How do you do? How do you get it into you? You absorb it. You absorb it. You have to absorb it. It has to go in here through what? What are the process absorptions? It has to go through the membrane, right? What are the processes? You know what they are. How do you get this food into the green part? Absorption. What is that? How do you transport across the membrane? Channels, carrier proteins, endocytosis, right? Right? Okay, so that's how we get stuff into the green part. So we need osmosis, diffusion, we need endocytosis. All that has to happen when we're eating, depending on what the material is. Glucose, for instance, needs a receptor, right? Needs a carrier protein. Ions like salt need ion channels, right? When you eat salt, don't you? It has to go into your body too. That's one of the things that has to go in. How does it get in there? Ion channels. Through what method? Diffusion. Does that make sense? This is all about diffusion and osmosis. Eating is all about diffusion and osmosis. There's some active transport too. There's some passive transport that we're discussing, but there's also active transport. Well, what do we know? What do we know we need when we're talking about this tube within a tube? If we want, is this enough folding? Well, one of the things, if you looked closely, if you took a close look at this intestine here, if we just kind of zoomed in on it, what we would find is the surface of the membrane does this. We call these microvilli. Microvilli are just even smaller folds. So we have folds upon folds upon folds. Why do we have all these folds in this membrane? So that we can get as much food, nutrients into the green part as we can get. Our whole point of our intestines is to get the food into you. You have to get the glucose, the fat, and the protein into your body. The only way that happens is through osmosis and diffusion. So we're a tube within a tube. We have to cross the membrane. We know all these are cells, right? All these tissues are made of cells. So you have to understand that this is a cell, this is a cell, this is a cell, this is a cell. You see how many cells there are? All these microvilli are all made of cells, all the way around. Do you see that? And why are they folding over and over again? Because that's going to increase the amount of time you have to absorb that food. You have to get that food into your blood. Of course, there's blood vessels in here too. Do you understand that? There's hepatic veins, hepatic arteries. All those blood vessels go to the liver, and that's what we call. This is a hepatic. Hepatic means liver, right? Hepatic artery, hepatic veins. All that blood goes around your intestines to try to get that food from outside your body, into your blood, so it can be transported. The blood transports the sugar, the proteins, the fats. Are we clear? All right. Yes? No, I want you to finish this unless it's an emergency. Question?
What causes you to throw up?
Huh?
What causes you to throw up?
Vomiting is caused by any kind of irritation to the stomach lining, or sometimes if your body gets an infection. So anytime you eat something that upsets your stomach here, right, it's going to vomit. It's going to make you vomit. It could be poison, your body reacting to poison. Irritants like hot stuff, spicy things are irritants. Spices are irritants. They irritate the surface of your skin. Your skin cells get irritated by them. It's almost like pepper spray. You ever been pepper sprayed in the eye? You ever seen people do it in movies and such? Well, when you pepper spray, that's what you're doing when you're putting hot sauce on your taco. That's what you do when you see somebody eating a habanero pepper. That's why they call it pepper spray. You're irritating your skin. No, please don't eat pepper spray. I'm just saying that's where we get the name. It's an irritant. It irritates. Peppers produce the pepper plants. They produce irritants to try to make animals stay away from them. Then we choose them because they have it, because we're crazy. But most animals that react the way we do that food, they go, nope, they're not going to touch that anymore. Now there's some animals like parrots that don't even have saliva glands, so they're not bothered by the peppers. But most of us are bothered by the peppers. Our eyes water. It hurts if you eat hot enough stuff, right? There's even challenges. We challenge each other to try to see how hot we can eat. They call them ghost peppers and such, right? Don't put things away. I got a couple more minutes. Let me finish what I'm saying. So, in any case, that's outside. That's folding. That's how folding helps us. That's called. Wait, before we put anything away, let me hit what's going to be on the test. You all want me to know that? You keep begging me to talk about it. There's one more thing that's also going to be on the test. There's something else. That's how we get food and water into us. How do we get, there's something else we need. What else do we need? Food and water, what else? Oxygen. How do we get oxygen? We have another tube, so that's one tube within a tube. Now there's another tube inside a tube as well. What's the other tube that's in that tube? It's also going through the mouth and nose, and it goes down, but it doesn't go all the way through us. It bifurcates or splits. Is the trachea splitting into what? Into the lungs. Right? Bad drawing, I know, but that's life. The left and right lungs, right? Left and right lungs. Right? Now, these tubes keep branching. Stop putting things away. They keep branching. They branch all throughout the lungs. If you opened up your lungs and you got rid of the connective tissue, you would see this amazing tree, all branched, one off the other. In a sec, it's time when I say, just a second. We're going to go ahead and look at this. And if you look inside here, what you'll see is a tube that ends like this. Look at it. And so this is all, air comes in here, goes through all these tubes, and ends up here. This is air. Air is made of what? Oxygen and carbon dioxide. Right? So this is called the alveoli. It's going to be on the test. Everything I'm saying is going to be on the test, so I'm not repeating it. Alveoli. Now the alveoli is there at the very end. It's a dead end to the lung. Around the alveoli are these blood vessels. There's arteries and veins throughout this. And there's an artery called the pulmonary artery that surrounds here. And I'm going to now I'm going to even go closer. I'm going to go even closer than that. I'm going to zoom in again. And right where the alveoli is here, and right next to it is this thing called the pulmonary artery. The pulmonary artery is the only artery that has low oxygen, high carbon dioxide. All the other arteries have high oxygen, right? That's right. And so in your lungs, in the alveoli, in that little space right here, right, in the alveoli, on the other side of that artery or vein, you have is high oxygen, right, and low carbon dioxide. So now let me ask you, where is the oxygen going to go? From where to where? From the alveoli into the blood. I hope you see that. And where's the carbon dioxide going to go? It's going to go out. And that's why you breathe. That's how you breathe, through diffusion, through simple diffusion. CO2 leaves the blood, it goes into the alveoli. Oxygen goes into the blood. through this simple diffusion, through a membrane. Again, what's in your lungs, when you're breathing in smoke, it stays in the alveoli. The problem is you start to get things that block the passage of air, which stops the oxygen from getting into your blood. All right, good luck. Okay, so when we're moving on and looking at membranes in general, the structure of membranes, a lot of organelles have membranes. Now, I want you to remember, I keep trying to put links between what we've learned before and what we know now. And we know that one of the difference between eukaryotic and prokaryotic organisms is that prokaryotic organisms have no membrane-bound organelles. So the organelles we're seeing here, these organelles are going to be found in eukaryotic organisms because prokaryotic organisms have no membrane-bound organelles. Now, the plasma membrane, of course, prokaryotic organisms have that because that's the membrane that separates outside from inside. But unlike prokaryotic organisms, especially multicellular eukaryotic organisms, have to have a way to connect their cells together, and that's what you see here along these lines. This is one membrane here of this cell, here's another membrane of this cell, and in between them you see these small dots, or you can think of them as welding points that hold the cells together. They're fusing points, if you will. So here you have a nuclear membrane that separates the nucleus inside from the cytoplasm outside. Here you have the mitochondria, which has an outer membrane, and then there's an inner membrane inside. So here's your outer membrane here, and then here's your inner membrane, the eye in here, and we'll talk more about that when we do respiration soon. Then here you have a Golgi body, and a Golgi body has another membrane, and in the membrane of the Golgi body, what you'll find is different sacs that allow for different chemical reactions to occur as the Golgi apparatus takes proteins from the endoplasmic reticulum and lipids from the endoplasmic reticulum and then gets them ready to be packaged out of the cell. So the Golgi apparatus is packaging and transporting things out of the cell. Then, of course, the plasma membrane separates outside the cell from inside the cell. And this is what a membrane looks like, and we have this idea of a fluid mosaic model. Now, think about what the word fluid means. Fluid, a lot of people think of fluid as a liquid. But remember, a liquid is only one type of fluid. All right, fluids, some examples of fluids are gases. So a gas is a fluid, and so is a liquid. So what is a fluid? A fluid is something that moves smoothly, right? So you have this, you've heard of fluid movement. So you have movement that flows like a liquid flows, like water flows. So the membrane flows, and what happens is in these membranes is these lipids, they dissolve each other. The nonpolar regions dissolve the nonpolar regions, and these lipids will flip and they'll move from one side of the membrane of the cell to the other, and they're constantly moving back and forth. One of the things that helps this movement along to keep its fluid, the membrane, to keep the membrane's fluid nature, is cholesterol. So cholesterol is very necessary for the membranes, the cell membrane, to keep its fluid nature. In other words, to keep it moving. It's important that this membrane be able to move, that this protein be able to move from here to some other place in the cell, etc., and just keep a constant interchanging of its components. A mosaic is the word mosaic that you see here is a word that represents one thing made of many things. So here you have this one thing, a picture of Barack Obama. And if you take a close look at it, it's made of many, many pictures of different people. So this is called a mosaic because it's one thing made of many things. So when we're looking at the plasma membrane, all right, what we're seeing is that the plasma membrane is made of many things, but itself is one structure. So even though it's made of phospholipids and carrier proteins and channel proteins and cholesterol and these things called glycolipids, and glyco means sugar, sugar lipids and sugar proteins, these glycoproteins, even though that's made of all these different kinds of things, that's why we call it that is why we call it a mosaic, because it's made of all these different components, but is actually one structure. Glycoproteins are proteins that have sugar on them, see the carbohydrate, right? Carrier protein we know are proteins that take a specific molecule from one side of the cell to the other. And this is a form of facilitated diffusion because you don't see any ATP and it's not dependent on a concentration gradient, for instance. You see that you have phospholipids, of course, you have cholesterol, you have a channel protein. Here you have, what is this? This is a carrier protein that does what? Look at this, what's happening here. ATP is being used. It's active transport, right? So this is the sodium-potassium pump. So sodium's is binding here and leaving, and then potassium would come in. Here's a phospholipid, a phospholipid with a sugar component on it. We call it glycolipids. So glycoproteins and glycolipids have these sugars on them. They act to stabilize the structure of the membrane, so like anchor points. So the fluid, you don't want the membrane to be too fluid, but you don't want it to be too stiff. You want to be able to bend and twist. I mean, could you imagine what would happen if your membranes weren't flexible? What would happen if you fell? You'd shatter as if you were made of glass. And so you don't want that. You want this membrane to be able to move with force, be able to flow. So that's a lipid, and that's the fluid mosaic model of a membrane. And now let's look at the questions. Identify the components of the plasma membrane involved in facilitated diffusion. Well, facilitated diffusion, what are we talking about? We're talking about channel proteins, and we're talking about carrier proteins, right? Those are the two types of proteins that you see here and the two that we've talked about in class that have to do about channel and carrier proteins. Active transport, you're talking about carrier proteins. Here it is. All right. Cell signaling. Well, what's gonna tell the cell to that something's happening outside? It has to be something that sticks outside and also is able to match a signal outside and transmit that signal into the cell to tell the cell that something's happening outside it. So how does your body know that the blood that your blood has too much sugar or too little sugar? It's these cell signaling proteins and that help with this. And the types of cell signaling molecules are glycoproteins and glycolipids. The glycolipids and glycoproteins can also be important in identifying a cell-to-cell recognition. So how do your cells know that you're you and not some bacterial invader? Why is it that some people reject organs? It's because these things that stick outside the cell, like a glycolipid and a glycoprotein, are used to recognize a cell as being part of that organism. Now we have regulating membrane fluidity. What helps with increasing or decreasing cell fluidity? That's cholesterol. The more cholesterol you have in there, the more fluid the membrane. The less cholesterol, the more stiff the membrane. So cholesterol is the answer there. And describe the fluid mosaic model of membrane structure. That's this idea that many components move within a sea of phospholipids. Constantly changing position within that phospholipid layer. Also, you should pay attention to this, that you see water, the H2O, there's oxygen and two hydrogens, so you can think of it as Mickey or Minnie Mouse, kind of going right through the lipid layer through diffusion, or in this case osmosis, right? And you'll also see here the CO2 going from via the concentration gradient, or from high to low. So CO2 is moving, water's moving, so osmosis and diffusion are happening through the membrane. Also, you can see the phosphate head is hydrophilic, the fatty acid tail is hydrophobic, just as we've described and labeled earlier. Let's move on. So, explain how the fluid mosaic model accounts for observed properties of cell membranes. Well, one of the things that you'll see in cell membranes is that membrane proteins or components are not always all right. So when the membrane components, when we're talking about explain how the mosaic model accounts for observed properties of cellular membranes, we know that membranes are fluid. We know that they don't always have the same composition. We know that the membranes, we can find a channel in one place one day and look for that channel later and it's somewhere else. We can do this with the fluorescent microscopy. We can see the different places in the membrane glowing in different times of the day. We know that certain membranes change in their composition, how much cholesterol they have, how much protein they have, etc. So in order to explain this and all the different observations we've made, we say that we can say that the membrane is composed of the membrane components are not always found in the same place at the same time, and we know that the membranes change in their composition from season to season. Membrane is constantly changing, and because of that, the way we explain it is that there's this phospholipid bilayer. That's also one of the observations we make, is that the membranes are made of a lot of phospholipids, and we know that there's a hydrophobic portion, the tails, and we know there's a hydrophilic portion, the heads, and they form the heads. The hydrophilic heads point towards water, and water is a solvent on both sides. And these proteins float like rafts inside the membrane, where the hydrophobic parts of the protein are embedded in the membrane, while the hydrophilic parts of the protein stick out as the protein folds in order to get away from water and be dissolved by the various phospholipid tails. And so what we get is this fluid, ever-changing, ever-moving membrane. So what I wrote here were the actual some of the observations rather than writing down the how the fluid mosaic model explains the observations. All right, discuss the various functional roles of membranes in cells. Membranes separate one space from another, right? We know that. And membranes help with identifying cells, selecting what goes into and out of the cell. So that's important. Name the cellular organelle that possesses a membrane. Well, there's a bunch of them, and you can choose one. I chose a bunch just to give you some ideas of how many there are, but there's nucleus, chloroplast, mitochondria, Golgi. Don't forget vesicles and both the contractile vesicles, food vacuoles, lysosomes. So a lot of cellular organelles have membranes. They separate spaces where different functions can happen. Describe the membrane's purpose of the organelle, for this organelle, the membrane's purpose in this organelle. Well, let's take a look at the nucleus. The main purpose of the nuclear membrane is to separate the DNA from the rest of the cell. It also allows mRNA through. It also allows calcium and other ions that act as signaling molecules that turn genes on and off. So the membrane does a very important job. It keeps our information that makes us who we are secure. Describe the purpose of cholesterol in the plasma membrane. It increases the membrane fluidity, so it allows the membrane to keep moving back and forth and makes it more resistant to physical forces, right? So, imagine, I know a lot of you have phones that you've dropped and they've been smashed. Imagine if that screen were flexible, so that when you drop the phone, it didn't shatter. Instead, bounced around like some Jello or like a piece of thin plastic. Wouldn't that be better for your phone? Well, it's better for you too. Your cells, instead of shattering when they come into physical contact, they're able to flex, to absorb the energy and move and resist disruption. They're also able to interchange the pieces and parts that make up that mosaic, that really complicated cell surface. So suggests why marine organisms in polar regions have very high proportion of cholesterol in their membranes. This is very interesting because there are some mammals that actually during the winter when it gets cold, mammals will often increase the amount of cholesterol in their membranes as well. Now, if you think of marine organisms, that's ocean organisms living in the ocean, in polar regions, so it's very cold. Why would they need a lot of cholesterol in their membranes? Because it's very cold, and they want to resist freezing. And one of the things, what's the difference between something that's frozen and something that's not frozen? It's the things that are not frozen are more fluid, right? They move better, right? So cholesterol helps that membrane from helps resist that membrane resist freezing, resist becoming stiff. If the membrane becomes stiff, it can't function very well. The protein can't move to allow sodium in, for instance. It becomes rigid, and it can shatter or break more easily. In fact, one of the ways that we open up membranes, separate the one phospholipid layer from another, is we freeze it, and then we hit it with a very sharp razor called a microtome. And when we hit it with a microtome, the membrane shatters, and we're able to separate the two layers. Well, we don't want that to happen to our membranes, so we keep it fluid. That's what life does. Suggest why list 3 substances that need to be transported in all kinds of animal cells. That's in the animal cells, glucose, proteins, nucleic acids, oxygen, and the list goes on, right? Animal cells need sodium, they need potassium, they need iron. All your vitamins and minerals all need to get in and out of the cell, or into the cell anyways. List 2 substances that need to be transported out of all kinds of animal cells. Well, carbon dioxide has to come out. Nitrogenous waste, like urea, has to come out, right? and draw the membrane. Well, I sketched this one together. You have a really complete one on the previous page. You've made a three-dimensional model of it. I haven't labeled it because, except for cholesterol, because you should be able to label all these. So I'm not expecting that you're going to have many problems doing this. So what's the role of membranes in cells? Hopefully you know that we have when we're talking about the ER, where we have lipid synthesis, so we produce lipids in the ER, isolation of enzymes, right? Membrane-bound lysosomes, they have enzymes inside those lysosomes that would tear you apart, peroxisomes and such that would break down your cell. You want to keep those enzymes away from everything else in the cell, so that's why we have a membrane. Again, one of the big picture function of a membrane is to separate spaces. Containment of DNA, the nuclear membrane, is one of the reasons we have a nuclear membrane, is to separate the nucleus inside the nucleus from outside the nucleus. You'll find a lot of the answers to the questions we just went through are on this slide, or on this page. Your role of the role of protein in membrane synthesis, right? So some protein synthesis occurs on free ribosomes, but much occurs on ribosomes bound to the endoplasmic reticulum, rough endoplasmic reticulum, right? And of course, the rough endoplasmic reticulum is also involved in proteins and phospholipids by adding proteins and phospholipids to that membrane, to the plasma membrane, right? So we have, this is the ER. Notice it's connected to the nucleus. It's going to become important a little later. So understand that that the rough ER makes lipids, makes proteins, right? And here, and we look at the plasma membrane, may take up fluid or solid material and form membrane-bound vesicles, right? So as soon as it takes it in, if there's the membrane-bound vesicle. We looked at a little movie on it in class. As the food comes in, it's called endocytosis. And then when it's a bigger form, when we're talking about a larger amount, we call it phagocytosis. But in any case, you have this stuff that creates a membrane inside that membrane that used to be part of the plasma membrane is now the food that this cell just took in. The Golgi apparatus packages the proteins and lipids for secretion, hormones, etc. Right? So if things are going out of the cell, this Golgi apparatus takes the proteins and lipids made in the endoplasmic reticulum, and it packages them up and gets them ready for fusion with the plasma membrane, then out into the area outside the cell. Communication and cell recognition. So how do you know this cell is part of you and not some other organism, some invader? On the cell surface, you have glycoproteins and glycolipids that signal, hey, this is me. Don't kill me. Don't attack me. But you'll also notice that there's things like cell signaling. So if one cell wants to signal this cell to do something, it's those glycoproteins and glycolipids that react to that. And of course, transferring energy. So lipids inside phospholipid bilayers inside the mitochondria and the chloroplast separate areas and allow for concentration gradients of hydrogen ions that we will use later when we discuss the formation of ATP. That's how mitochondria is able to make ATP, because of all those membranes inside the mitochondria. But this is the same thing happens in chloroplasts. So if we take a good close look at the mitochondria, this is their matrix, and what you see, and we'll talk about this, we're going to break it up, so don't worry too much about this. This is just an example of how the membrane can be used. You'll see that you'll have folds inside the membrane that increase surface area, but also create different sections. You have this space here, and then you have this space here. And because you have two different spaces separated by a membrane, you're able to have two different concentrations. If you have two different concentrations, now you can have flow in one direction. Remember, we call that indirect, right? Indirect processing of energy, where indirect active transport. When we move things from one side to the other through diffusion, that facilitate diffusion, that can drive other processes, and that's how we make ATP in the mitochondria. All right, so discuss the importance of membrane systems and organelles providing compartments within the cell. So here we talked about all these different compartments and all these different functions. You have to read this. You can't just go by what I'm saying. I'm just summarizing this page. You have to actually read it. Now consider this. Consider that all these different processes, all the digestive enzymes in the lysosomes, the food being separated from the rest of the cell, all the different processes that are happening in the Golgi body versus the endoplasmic reticulum. Consider this. All these different processes happen in different spaces because the conditions in the spaces are different. For example, your bedroom can be a bedroom because it's dry. Your bathroom can be a bathroom because it has a sink and a bathtub that is wet, there's water running. So each space has its own function, and it can have that function because of the space, the walls that surround it. So because you've created these separate spaces in the endoplasmic reticulum, in the Golgi body, in the food vacuole, you're able to separate the functions that happen in each of these compartments. Just like that big house and little house we talked about earlier when we months ago when we started talking about the cell. When you create these small compartments, you create regions where different chemical reactions can occur separate from all the other chemical reactions. Still one cell, but different chemical reactions can happen in each of the different spaces. So that's what's important about providing different compartments inside the cell. You can have much more complex reactions, much more complicated chemistry going on than you can in a simple bacterial cell that only has one space. Remember, a bacteria only has one space. Sure, it has ribosomes, it has different chemical reactions, it has DNA. All that's true. It has small plasmids. But this is one space. That's that simple shack where the other eukaryotic cell has many different organelles, membrane-bound organelles, that allow the organelles to be separated, have different spaces that are separated from one another and allowing for different processes. Okay, so that should help you with this, and hopefully you understand, and we'll talk about it in class on Monday. Okay, so I think you can figure out what's going on with the structure of the membrane and what we thought the membrane was like before. We knew before we started to use this technique, we knew that the membrane was made of proteins and phospholipids, but we didn't know the structure of it. And what we thought was going on is you had this phospholipid bilayer, or this phospholipid layer, either way. All right, phospholipid bilayer. And then on top of that, you had like a protein coat, almost like a peanut M&M, reverse peanut M&M. So you had this phospholipid bilayer, and then on the outside of the lipid bilayer, you had this protein coat all the way around. So that made sense to people. For whatever reason, there was evidence to support that. So then, in order to verify that or to disprove that, what scientists thought was, well, if this is true, that this is the membrane in the cell, then we should be able to freeze this thing solid, and you can read the instruction, the paragraphs here and try to figure out what they mean. Freeze it solid and break it. So freeze it solid and then break it with a razor blade. And then take that little piece that's been broken and look at it underneath a microscope. So we should be able to split these two layers apart. Well, when we did it, this is what we got. We got this really you know, if you thought, if it were true that this was the model for that this model was correct, that you had a protein layer outside a phospholipid layer, when you split it, what you'd expect to see is, well, a smooth protein layer and then a smooth phospholipid layer. That's what you'd expect to see, but you don't see that. And if you think about the function of the cell, that wouldn't really work with function either, because how could you get stuff through that phospholipid layer, right? That's one of the things that could help you answer give a more detailed answer to one of the questions from the previous page. Because how could you get things like sodium ion into the cell if all you had was a protein coat on the outside and the phospholipid inside? There's no openings in the cell. So it's the key here is those openings into the cell and out of the cell to help things get in and out. This model didn't explain it, right? This theory of proteins coating the phospholipid layer just didn't work. And then when we took some electron micrographs after we split it, we saw a bunch of bumps on the inside. So we saw what we saw was this kind of like bumps coming out like this, the membrane layer, and there were holes through the membrane layer, and now the proteins that were here, you can see that they would fit. They were in the other layer. If you looked at the other layer would look something like from the bump, right? And then, oops, it would look something like something like this. And so they matched. But what were those bumps? What were these bumps and these holes? Those were the proteins that were embedded in the membrane. The proteins didn't coat the membrane. They were embedded in the membrane. They weren't some kind of candy shell on some peanut. They were the doors and windows into and out of the cell. So how did this show that this model, this protein coating the membrane, was the phospholipid layer? How did this show it to be flawed? Because as you have bumps and holes, it shows that the proteins are embedded in the phospholipid layer and not coating the phospholipid layer. It's just a fairly simple answer. So hopefully you have a better understanding of the lipid of the membranes. I think we've addressed a lot of the issues that people had with organelles. Please know that you have to understand what the organelles are and what they do, and the importance of membranes and the difference between prokaryotic, eukaryotic cells, plants and animal cells, and protists and prokaryotes, which are bacteria, right? The difference between prokaryotes and eukaryotes. So know that you have to know these differences. It will be on the final. This is something you're going to have to keep up with. All right, have a good day.